A quick note, this story is shared purely for educational purposes. Experimental treatments like this carry serious medical, legal, and safety risks. Please always work with a licensed veterinarian when it comes to your dog's health. It's 2 a.m. in a Sydney apartment. A man is sitting at his laptop. His dog is asleep on the floor beside him. She has tumors growing across her body, and a veterinarian has told him she has 1 to 6 months to live. He types 7 words into ChatGPT. How might I cure mast cell cancer in dogs? He's not a doctor, not a biologist, not a veterinarian. He's an electrical engineer who builds machine learning systems for banks and supermarkets. He's never studied a single page of oncology in his life. Within months, he'll use artificial intelligence to design a personalized mRNA cancer vaccine for his dying dog, and it will work. If you've ever sat in a vet's office and heard the words, "There's nothing more we can do," hit subscribe right now, because this story is about what happened when one man refused to accept that sentence. His name is Paul Conyngham. He's 42 years old. The dog on the floor beside him is named Rosie, a Staffordshire Bull Terrier Shar-Pei cross he adopted from a Sydney shelter in 2019. She came into his life during one of his hardest chapters, a breakup, difficult business decisions, long walks through the forest where neither of them needed to say anything. Paul called her his best mate. He meant it the way dog people always mean it, completely. Rosie was energy and joy packed into a black coat. She loved chasing rabbits more than anything in the world. Remember that. Here's a detail that makes the rest of this story even more cruel. Shar-Peis are 26 times more likely to develop aggressive mast cell cancer than the average dog. They carry more mast cells in their tissue. Their tumors appear earlier and spread faster. Rosie's breed mix did not just make her lovable, it made her a genetic time bomb for the exact cancer that would one day find her. Around 2022, unusual lumps appeared on her head. A vet dismissed them as harmless warts. Nearly a year was lost. By 2024, Rosie was formally diagnosed with late-stage mast cell cancer, large tumors on her hind leg, 1 to 6 months to live. Paul tried everything conventional medicine could offer: multiple surgeries, chemotherapy, immunotherapy, tens of thousands of dollars spent trying to buy time. The disease slowed. It did not stop. And eventually a vet looked at Paul the way vets do when they've run out of options. Then came the night with the laptop. Rosie was running out of time, and Paul did the only thing he knew how to do. ChatGPT did not hand him a cure. It handed him a plan. A 5-stage pipeline that read like something from a pharmaceutical company's research division. Sequence Rosie's DNA. Compare her healthy cells to her cancer cells. Use AI to model the mutated proteins in 3 dimensions. Identify targets the immune system could recognize. Design a vaccine. Paul is not a biologist, but he is a data scientist with 17 years of experience in machine learning. When the genomics director at the University of New South Wales questioned whether a private individual could handle 350 gigabytes of raw genetic data, Paul told him, "No worries. I'm a data analyst, and I'll figure this out with the help of ChatGPT." He did. Using pipelines he built with AI, Paul compared Rosie's healthy DNA to her tumor DNA and found the mutation driving her cancer, a gene called c-KIT, permanently switched on, telling her cells to divide without stopping. Then he used AlphaFold, the protein structure tool from Google DeepMind that won its creators the Nobel Prize in Chemistry just weeks before Paul opened it on his laptop, to map the mutated protein in 3 dimensions. The genomics director, Martin Smith, later described the moment he saw what Paul had done. It was like, holy crap, it worked. Coming up, the pharma company that said no, the paperwork that took longer than designing the vaccine, and a moment at a dog park that made it all worth it. Paul found a promising immunotherapy drug that could help Rosie. He contacted the manufacturer and requested compassionate use. They refused. The drug was for humans, not dogs, so he built his own. His algorithms identified which of Rosie's mutated proteins would most likely trigger an immune response. These are called neoantigens, proteins unique to her cancer that her immune system could be taught to hunt. The output was half a page of formulas describing a custom mRNA sequence. Professor Pal Thordarson at the UNSW RNA Institute agreed to manufacture it. His lab synthesized the mRNA and encased it in lipid nanoparticles, the same delivery technology behind the COVID vaccines. The entire process took under 2 months. But here's where the story turns maddening. The vaccine was ready, Rosie was dying, and no one at the university could legally inject it. Paul spent the next 3 months writing a 100-page ethics document, 2 hours every night after work. He later said the bureaucratic hurdles were actually tougher than creating the vaccine. And even after all of that, the university still could not administer the shot. It took a connection through Mari Madej, founder at the Canine Cancer Alliance, to link Paul with Professor Rachel Alavina at the University of Queensland. Alavina already held ethics approvals for experimental canine treatments. She could do what no one else had been willing to. In December 2025, Paul put Rosie in the car and drove 10 hours from Sydney to Gatton, Queensland. 10 hours with a dying dog in the passenger seat and a vaccine in a cooler. In a university veterinarian lab, Rosie received her first injection. Then he waited. Within 1 week, the tumors began to shrink, visibly. One tumor shrank by 75%, another by roughly half. Rosie's energy came flooding back, her coat turned glossy. She started moving like a dog who had forgotten she was supposed to be dying. And then came the moment. 6 weeks after treatment, Rosie was at the dog park. She spotted a rabbit on the other side of a fence and she jumped it. A dog who had been handed a death sentence launched herself over a fence because she saw something worth chasing. That is the image this story leaves you with. But this story is honest. So here's what also needs to be said. Not every tumor responded. At least one large mass did not shrink at all. The University of New South Wales published a statement in March 2026 saying clearly that Rosie still has cancer and it is still incurable. Paul is already resequencing the resistant cells to design a second vaccine. Scientists have raised legitimate questions about sample size, confounding treatments, and the absence of peer-reviewed data. All of that is true. But here's what's also true. A man with no biology training using freely available AI tools and a $3,000 sequencing bill replicated the core pipeline of pharmaceutical programs that cost hundreds of millions of dollars and take years to complete. Moderna and Merck are running clinical trials right now on nearly identical personalized mRNA cancer vaccines for humans. Their first approvals are not expected until 2027. Paul did it in months, from his apartment, for his dog. Professor Thordarson called it the first time a personalized cancer vaccine has ever been designed for a dog. Then he asked the question that hangs over this entire story. If we can do this for a dog, why are we not rolling this out to humans? AI did not cure Rosie's cancer, but it turned a grieving dog owner into a functional cancer researcher in a matter of months. And somewhere in that transformation is something that matters to every person who's ever loved a dog and been told there was nothing left to try. Paul is still at his laptop working on the next vaccine, and Rosie is still here, still chasing rabbits, still jumping fences, because that's what happens when the person who loves you most refuses to let the world tell them it's over. Which part of this story hit you the hardest? Tell us in the comments. This has been Happy Tales. If you want to discover more incredible stories about the dog on your couch, watch this video next. See you there.